Now at this time, let's select 3 different fluorophores and 3 PMTs, and we'll look at our separate images. So I'm going to click on the Leica settings, and I'm going to go to Fitzy Tritzy CY5. This is now 3 fluorophores that we're going to be exciting based on 3 laser lines. I'm going to turn off the UV laser line since we don't need that. But you see a 488 laser line, you see a 543 laser line, and you see a 633 laser line, all illuminated on our spectrum. You also see the various emission curves as a result of the excitation based on these laser lines. And concurrently, we have a photomultiplier tube set below this graph of where the emission is going to take place. So this photomultiplier tube is collecting here, this PMT is collecting here, and this PMT is collecting here. We're going to be outputting our image in 3 colors, green, red, and blue. But we can also modify these colors, as you'll see in a second. Also, please note that all 3 photomultiplier tubes are now activated. So I'm going to click on Live, and if you look on the right screen, you'll now see an image with 4 windows in it. The first one is the Fitzy, the second one is the Tritzy, and this is the CY5. Again, we want to set our smart gain and offset for each of these windows. So we'll click on the first one. If you double click, only that one comes up. And now we can increase our smart gain. Again, we want to be around 900, maybe 950, certainly below 1,000. So we're at 978 on our smart gain. Let's check our offset. Our offset should be minus about 0.3, 0.4. Remember, we can always use the lookup table to show us our green and blue pixels. So if we lower the smart gain, make some of our blue pixels go away. So we have few blue pixels, and use our smart offset until the green pixels just go away. Now we have a perfectly set image. We'll click through our lookup table, and now our Fitzy is set perfectly. If we double click again on the Fitzy window, the other windows come up again. Let's double click on the Tritzy window. And now we'll set our smart gain and our offset. We'll go to lookup table first. Too many blue pixels, so we'll lower our gain until our blue pixels start to go away. And we'll control our offset until our green pixels appear and then go away. So right about there. So we'll double click again on the lookup table. Now our Tritzy looks very nice. We'll double click on that window, and now we're in the CY5 window. Again, we'll click on the lookup table. We'll get rid of our blue pixels. You can see that there's a lot of blue pixels there. So we'll minimize those, and now control our offset. That looks pretty good. Double click on that window, and now we'll click through the lookup table, and we see the composite image showing all of this information output into one different image. We have 3 distinct colors here. We have a blue color, a pink color, and then a golden color, actually a 4th color, sort of a brownish color. So we're really getting a lot of information back by engaging all of these PMTs. Also, don't forget that your focus is very important. You could be under-focused or over-focused, and that will dramatically impact the quality of your image. As you can see, I'm focusing through back and forth, and you can see how the focus can actually eliminate the illumination altogether. So make sure that you're highly focused on your specimen. One easy way to do this is to use the transmitted light PMT. So we'll double click on the composite image. We're going to go over here and click on stop. Let's deactivate all 3 of the photomultiplier tubes, and we'll turn on only the bright field photomultiplier tube for transmitted light. Now if I click on live, here is my image in bright field. Again, I can use my lookup table to look at blue pixels, minimize those, and green pixels. get a few green pixels, and now double click through my lookup table, and now I have a decent transmitted light image. But notice how it changes dramatically when I focus back and forth through the specimen. Transmitted light is returning more information to you about the specimen than the fluorophores are. So your focus is very critical, both on transmitted light and in fluorescence. There is also the possibility that I may want to use all 4 PMTs simultaneously. I might have 3 different fluorophores that I'm looking at, and I also want to use the transmitted light PMT, which we already used. So let's turn them all on and look at our image. So I'm going to activate the PMTs. All 4 are now activated, including the transmitted light PMT. We'll click on Live, and now if you look at the right screen, you see all 4 of our PMTs illuminated together with our composite image. Now, why is this important? Sometimes there are structures that can only be seen in transmitted light. For example, that nucleus there's a nucleus down here inside of this cell. There's also other cellular material in here that you can see that is not illuminated by any of the fluorophores. For example, if you look at the FITC, you see no nucleus here. You see some of this intracellular material here, but you don't see that nucleus, which is a region of interest. What about the TRITSI? Same thing, no nucleus. What about the CY5? Once again, no nucleus. But if I go to the transmitted light PMT, voila, there's my nucleus. So sometimes there are structures that you need to see present in your specimen that will not be illuminated by a fluorophore, and so it's important sometimes to use all 4 PMTs. Then you get a true picture of everything that's going on in your specimen. Now that we've selected our images and made some images and corrected them based on the smart gain and offset using our lookup table, let's concentrate on some other tabs under the acquisition mode that allow us to capture different kinds of images. Up till now, we've been doing XYZ. XYZ allows us to capture images in X and in Y and in Z, so we can do Z stacks, which we'll come to in a few minutes. But there are also other features available to us here. One very important one is XY lambda. XY lambda allows us to capture images in X and Y according to different wavelengths. So we can actually turn on and turn off different wavelengths as we're capturing our XY images. So that's very useful. Also useful is XYT. What is XYT? XYT allows you to do imaging in a time-lapse mode. You can collect your images in X and Y according to a time that you set up in hours, minutes, seconds, and milliseconds. So now you're actually creating movies of your cellular processes occurring in real time and storing that information as movies on the computer. So XYT is very useful. You can also do XY lambda T. Again, there are several functions that allow you to capture images in X, Y, Z, in time, and in lambda. So the system is very flexible when it comes to acquisition of images. Also on this tab, let's switch back to XYZ, is the Sequential Tab. And this allows you to select how you're going to scan your images. You can scan between lines, which means as you image a line across your image, as you scan that line, you can drop down and scan between the line before you start another line. So this will augment the amount of light information that you're collecting. You can also sequentially scan between frames. So after every frame is taken, you can go back and select yet another frame and then continue. You can also scan between stacks. A Z-stack is a collection of images that are shot through a vertical portion of your tissue. We'll come to Z-stacks in a minute. So sequential scan is very useful. Also, we have what's called tile scan. What is tile scan? You can tell the computer to go to a particular tile on the position of the stage. So let's say in your specimen, you want to set up 3 or 4 different tiles. You can tell the system, in this tile, I want to scan XYZ. In this tile, I want to scan XYT. In this tile, I want to scan XY lambda. So again, it gives you the power of the motorized stage and the motorized focus to get as much information as possible from each tile across your specimen. There's also what we call mark and find. Mark and find allows you to mark a region of interest that you can go back to later. So if during your setup and preparation you find portions of your specimen that are of interest to you, you can mark those in the computer and the system will go back to them on demand at a later time. Finally, on the acquisition mode tab, you have a best focus panel. I don't particularly recommend the best focus panel because you're depending on the computer to decide what the best focus is for the microscope. I think it's better just to find the best focus yourself. You know your tissues better than the system does, so just rely on your own eyes to tell you what is best focus. Now let's finish discussing the other options that are available to us under the XY tab. We've already discussed resolution. We will, in a few minutes, go back and select some very high resolution images, and we will do line averaging and frame averaging to get the better images from these resolutions. So we'll come back to that in a second. For now, let's leave it on 512 by 512. We also have a pinhole feature that allows us to change the size of the pinhole, and I'm going to show you how that affects your image in a second. Remember, the pinhole in the microscope is used to eliminate extra light that is being produced as a result of the laser excitation. The laser excites all the fluorophore that is present throughout your tissue. If you allow all that light to come back to the observer or back to the camera, it will be too much information and you will not be able to resolve your structures, let alone the molecules that you're interested in watching in live processes. So therefore, it's important to use a pinhole to eliminate that stray light and give us a nice clean optical section of the image that we're looking at. The system is always defaulting to a standard pinhole that gives optimum results for the objective used, the fluorophores used, etc. But as is everything else in this software, the pinhole can be manipulated. It's not recommended that you play with the pinhole, but in some instances where you do not have enough illumination returning to the camera, you may want to widen the pinhole. And so if you look down here at the bottom of this tab that opens up when you click on the pinhole, that opens up for you, and now you can change the size of your pinhole. The smallest size of the pinhole is down around 20 to 30 microns in diameter, but you can increase that all the way up to over 500 microns, which is certainly not recommended. This would be no better than a standard fluorescent microscope. The whole point of the pinhole, and there's actually 2 pinholes, one that you can manipulate right after the illuminator, and another one that's inside the microscope, which you cannot manipulate. The whole point of the pinholes is to give you the sharpest high-resolution image as possible. We will play with the pinhole a little bit, and I'll show you the difference in the illumination when we do that. Note also that the system gives you the ability to select the size of your pinhole in microns or in Airy units. We're not going to take the time to discuss Airy units right now. Basically, one Airy unit is comparable to about 61 microns. And that's sort of a standard that's selected in the industry for resolution. But realize that your pinhole can go all the way down to about 30 microns and all the way up to about 500, which we discussed before.